Hello students, today I am going to talk about what urine formation. So this urine formation, it takes place in a three steps. There are three steps involved in a urine formation. So what are the three steps? Let's see one by one. The first step is called as the glomerular filtration. Glomerular filtration is also called as what? Ultrafiltration. And the second step is what? Reabsorption. And third step is called as what? Secretion. So urine formation takes place in a three step. First is glomerular filtration, also called as the ultrafiltration, reabsorption and secretion. Let's see one by one what are the how it takes place. First we will talk about what glomerular filtration. Glomerular filtration. So actually glomerular filtration in this, first of all we have to understand some basic terminology. Suppose if this is the glomerulus, this is what afferent arteriole. Afferent arteriole. Why we are calling it as afferent arteriole? Because first of all, afferent arteriole is arising from one artery. The name of that artery is called as what? Renal artery. So renal artery gives rise to what? Afferent arteriole. Afferent arteriole, from arteriole always capillary will form. See, from aorta. Okay, this is what? Descending abdominal aorta. From there, renal artery will arise from that afferent arteriole. Why afferent? Because afferent means bringing toward organ. Efferent means away. Okay, so it is bringing the blood to glomerulus. And from the glomerulus, the blood is carried away by efferent arteriole. Efferent arteriole. I think up to here you understand. Now, what is happening here? Below this glomerulus, what is present? Bowman capsule is present. Okay, and this portion is called as what? Lumen of Bowman's capsule. Lumens of Bowman's capsule. Clear? Acha. Like that, the next part is PCT, DCT. Okay, collecting duct, many things are there. Loop of Henle. This is distal convoluted tubule, and this portion is called as what? Collecting duct. This portion is called as distal convoluted tubule. This portion is called as proximal convoluted tubule. Proximal means what? Near. Convoluted means what? There is one plane surface is there and one convoluted surface. So here this tube is very convoluted. Why this tube is very convoluted? And it is near to the glomerulus, that's why proximal means near. It is at distance from this glomerulus, so it is called as distal, distance. Clear? So wherever this convoluted surface you will see, there you will find it will increase the surface area for absorption. Now what will happen? Glomerular filtration takes place. So when the blood will come here, the blood has to cross what? Blood capillary. Okay, and after that, here basement membrane. Okay, here what is present? Basement membrane is present. What is this? Basement membrane. And here this portion is called as what? Epithelium of Bowman capsule. Epithelium of Bowman capsule. Okay, so the urine will filter out through these three membranes. Okay, so how it is? Let's see. First of all, I am telling you how glomerular capillaries, who creates the blood pressure here. Actually in this, what is the glomerulus? It is the bunch of blood capillary. Okay, glomerulus is what? It is a bunch of blood capillary. In this, blood pressure is created, because of that blood comes out. Okay, now how it is taking place? Let's see. First of all, afferent arteriole here. This is what? Afferent arteriole. So afferent arteriole lumen is what? Somewhat broader. The afferent arteriole will convert into what? This is what? Afferent arteriole. And this portion is called as what? Glomerulus. And from the glomerulus, what will come out? Efferent arteriole. What is this? Efferent arteriole. Now one thing, the afferent arteriole lumen is broad. That means more quantity of blood is flowing here. But outlet of the blood, efferent arteriole, which is taking the blood away from glomerulus and it is bringing the blood toward the glomerulus. So this lumen is wider and this lumen is what? Narrower. Okay. So what will happen? Entry of blood is more but exit is what? Less. This creates here blood pressure in the blood capillary of glomerulus. Is that clear? So glomerular blood capillaries, blood pressure will be created and small quantity of blood will come out from this glomerular blood capillary. Okay, so if we will see the glomerular blood capillary, this is what? Glomerular blood capillary. Glomerular blood capillary. From here the blood is flowing. Achha. Because of very high blood pressure, actually the wall of blood capillary is having small holes called as what? Fenestra. The name of these holes are called as what? Fenestra. Okay. After this fenestra, there is one membrane is present here. The name of this membrane is called as what? Basement membrane. Okay, basement membrane is made up of proteoglycan. Okay, and actually this basement membrane is made up of what? Proteoglycan. And this basement membrane is what? Negatively charged. This basement membrane is negatively charged. Now what will happen after this basement membrane? What will come? This is what? Epithelium of Bowman capsule. Epithelium of Bowman's capsule means here blood will first come out from this glomerular blood capillary, then it will cross what? This basement membrane, and after that the blood has to cross what? This epithelium of Bowman capsule, and then here whatever liquid will be accumulated into the lumen of Bowman capsule, this liquid will be called as what? This liquid will be called as what? Filtrate. Okay, now let's see. So from here blood will come out. Okay, blood, and then it will cross this basement membrane. Okay, and after that it has to cross this epithelium of Bowman capsule. So one thing I want to tell you, that epithelium of Bowman capsule is made up of specialized kind of cells. The name of these cells, okay, a special kind of cells epithelium of Bowman capsule is consists of. The name of these cells are called as, okay, podocytes. Podocytes. They are called as what? Podocytes. Okay, and the gap between two podocytes, here you are seeing this gap. This gap is called as what? Filtration slit or slit pore. Filtration slit or slit pore. Filtration slit or slit pore. Okay, so these all cells are what? Podocyte. This podocyte is forming what? Epithelium of Bowman capsule. Now one thing, from here small quantity of blood will come out, and through this filtration slit it will come here. This part is called as what? Lumen of Bowman capsule. Okay, see, here it is coming and here lumen of Bowman capsule it is accumulating. So from here this water is coming and from here it is coming here. Okay, so the blood which is filter out from these three layer, what are these three layer? This is called as what? Tell me. Acha, the blood capillary wall is made up of what? Wall of blood capillary is made up of what? Endothelium. Endothelium is also called as what? Tunica intima. So endothelium or tunica intima, that is made up of what? Simple squamous epithelium. So simple squamous epithelium, I think you know. What is simple means what? Single layer. Squamous means what? Flat. Epithelium means name of tissue. So single layer of flat cell is called as what? Simple squamous epithelium. Okay, so when the blood is coming out from this fenestra, this is the glomerular blood capillary, it has to cross the basement membrane, which is a negatively charged, and through the filtration slit or slit pore. That means what? The gap between two podocytes, it comes and accumulate into the lumen of Bowman capsule. So here whatever the blood has filtered out and accumulated here, this blood is called as what? Filtrate. Acha, in this filtrate RBC is absent and plasma proteins are absent. What are the four plasma protein? Albumin, globulin, prothrombin, fibrinogen. These are absent in this filtrate. Okay, in this filtrate what are the things absent? RBC absent and plasma protein, albumin, globulin, prothrombin, fibrinogen. These all are also what? Absent. Okay, in this filtrate. So why there? Now the question is what? Why in this filtrate which is accumulating here into the lumen of Bowman capsule, in this RBC and plasma proteins are absent? The reason is what? RBC, WBC platelets. Okay, one thing I want to tell you, that RBC, WBC and platelets, these three are what? Negatively charged. Why they are negatively charged? Because their plasma membrane is having phospholipid bilayer, in that negatively charged is there. As well as if you talk about the plasma protein, so plasma protein is also having which charge? Negative charge. Okay, and here this basement membrane is also what? Negative charge. So if any RBC, WBC platelet or any plasma protein like albumin, globulin, prothrombin, fibrinogen, if they want to cross this basement membrane, they can't cross it. Why? Because basement membrane is also negatively charged. Negative will repel each other. So that's why in this filtrate RBC, WBC platelet and plasma proteins are what? Absent. But there is one condition is there, glomerulonephritis. Okay, glomerulonephritis. So wherever itis word is coming, okay, where, 1 minute students. Sorry for disturbance. See, wherever itis word is there, itis word is related to what? Pain or inflammation. Inflammation of glomerulus, it's inflammation. Okay, during glomerulonephritis, what is happening? This basement membrane lose its negative charge. So because of that, RBC or albumin, globulin, prothrombin, fibrinogen, that plasma protein can easily pass this basement membrane and they will come along with what? Filtrate. Okay, so that if albumin protein is coming into the filtrate and in the urine albumin protein is found, that condition is called as what? Albuminuria. Albuminuria. So albuminuria takes place at the time of glomerulonephritis. Basement membrane lose its negative charge. Albumin protein, which is a negatively charged protein, that can easily cross because no one is there to repel it. Okay, and it will also come into the filtrate. Okay, now first of all, these podocytes are arranged in intricate manner. Which manner? Intricate manner. Such that all the constituent of plasma except RBC and plasma protein come into the lumen of Bowman capsule. This process is called as what? Ultrafiltration. So ultrafiltration takes place because of what? Filtration slit or slit pores. Okay, so podocytes are arranged in an intricate manner and leaving a small gap in between them, that is called as filtration slit or slit pore. From there, when the plasma protein comes out, when the blood plasma comes out from this, are all the constituent of plasma except formed element RBC, WBC platelet and plasma protein, except these two things, everything will come here, okay, into the lumen of Bowman capsule. Is that clear? So this one is called as what? Ultrafiltration. Okay, in 1 minute kidney filters, in 1 minute kidney filters, okay, 1100 to 1200 ml of blood. So 1100 to 1200 ml of blood is filtered by the kidney in 1 minute. This is what? 1 by 5th of cardiac output. Cardiac output. So in 1 minute, the heart is pumping how much blood? The heart is pumping 5 liter of blood, and how much kidney is purifying, filtering the blood? That is 1100 to 1200. That is how much of the cardiac output? 1 by 5th of the blood pumped by the heart. Okay. Now, in 1 minute, how much filtrate accumulate here in the lumen of Bowman capsule? That is called as what? GFR. Next, we are going to talk about what? GFR, glomerular filtration rate. Glomerular filtration rate. So, what is this? Amount of filtrate, accumulate in lumen of Bowman capsule in 1 minute is called as what? GFR, glomerular filtration rate. So normal GFR is what? 125 ml in 1 minute, or you can also call it as what? 180 liter per day. This is the normal glomerular filtration rate. Very, very important for the Neet point of view. Okay, so this one. And now we are going to talk about some calculations. See, when the blood plasma or blood is crossing, first what it will cross? The blood which is flowing in the glomerulus will first of all what it will cross? It will cross what? Endothelium. It will cross endothelium. After that, the blood has to cross what? Basement membrane. And at last, the blood has to cross what? Epithelium of Bowman capsule. Epithelium of Bowman's capsule. So these three blood has to cross. So first of all, here a blood pressure is created in the glomerulus. That is called as what? Glomerular hydrostatic pressure. The blood pressure created in the glomerulus is called as what? Glomerular hydrostatic pressure. The value of glomerular hydrostatic pressure is what? 60 mmHg. So blood pressure in the glomerulus is called glomerular hydrostatic pressure. Okay. Now what is happening? Because of that, blood will come out. Means plasma will come out. Okay. Then in this blood capillary, one pulling pressure is also applying here. The name of that pulling pressure is called as what? BCOP, blood colloid osmotic pressure. Okay, so blood colloid osmotic pressure pull ups the blood and not allow the blood to flow out, whereas glomerular hydrostatic pressure push the blood downward, and blood colloidal osmotic pressure pull the blood upward. Clear? So it will range will be between maybe near about -28 to -32 mmHg. Okay. Acha. Now see, when the blood is crossing this epithelium of Bowman capsule, this epithelium of Bowman capsule is applying opposite force on the blood and not allowing the blood to enter. Okay, it is opposing this membrane. Epithelium of Bowman capsule. Epithelium of Bowman capsule is made up of what? This is also made up of what? Simple squamous epithelium. This is opposing the blood. So this pressure which is applied on upper side by this membrane is called as what? Capsular hydrostatic pressure. What we can write here? Capsular hydrostatic pressure. So epithelium of Bowman capsule is applying opposite pressure to the blood which want to come here and accumulate. Okay, capsular hydrostatic pressure value is -18 mmHg. Why I have written minus? Because it is opposing pressure. Okay, blood colloidal osmotic pressure, which is here, that is a pulling pressure. Okay, so effective filtration pressure, if we will talk about what effective filtration pressure is equal to glomerular hydrostatic pressure minus blood colloidal osmotic pressure plus capsular hydrostatic pressure. Okay, so glomerular hydrostatic pressure. So what is the value of glomerular hydrostatic pressure? It is how much? 60 mm Hg minus blood colloidal osmotic pressure. This we have to take. Okay, 28 we don't have to take here. So minus 32 plus minus 18. So 32 plus 18 how much? It will become 50 minus 50. So how much it is coming? 10 mm Hg. So with the pressure of 10 mm Hg, the filtrate or the blood comes into the lumen of Bowman capsule. This is called as what? Effective filtration pressure. Okay, I hope you understand this thing. Okay, so this was all about what? Glomerular filtration. In next lecture, I am going to talk about the reabsorption in the part 2. I hope you understand. Thank you.